Well, thank you for having me. I'm sorry for not being able to be in person. I'm in Madrid right now, and we'll be back in Canada this afternoon. But I'm very excited about this trial. I think it's a very innovative and important trial. And I'm happy to serve as the ECHO-Corelab director for this trial. As you know, I'm based in Quebec, so in somewhat still cold part of the world. If you go to the next slide, we're going to cover rapidly the agenda of this presentation. So first I would like to spend some time on how we operate as a Corelab, what is the workflow in the ECHO-Corelab. Then what are the objectives of the echo imaging protocol in the context specifically of the Rheia trial. Then we'll discuss, I don't want to spend too much time, but some of the main points regarding how to prepare examination, positioning, and general procedures. I guess that there are some study coordinators in the room, and they may be interested, to know some of the items regarding the Corelab and the image acquisition sequence. I won't go through the complete image acquisition sequence. There will be a more detailed PowerPoint and webinar to be provided later. So, and finally, how to transfer the echoes to the ECHO-Corelab from the site to the ECHO-Corelab. That will be the last point. But this may change because it will depend very much, the protocol will depend very much on which company will be used to do this image transfer. So if we go to the next slide, that should be, you should see a busy algorithm with yellow and blue and green boxes. So the way we work, we are connected to all the participating sites, and the echoes are transferred from the site to the ECHO-Corelab, to our server in the ECHO-Corelab, using a company, an image transfer provider. We've used so far in the other partner trials that we've done in the past and still doing, an early TAVR and TAVR unload. We use the Bioclinica. I think for this trial we may use a different company, but this is not that important. I mean, the important way is that the echoes are transferred efficiently and not corrupted to the Corelab. So that is the first step. And then the second step is we have an initial quality assessment of the echo to make sure that the echoes are complete, well anonymized, and then they are dispatched to our sonographer. We work as batch, so we do generally a batch of 12, 15 echoes per batch, and they are transferred to the sonographer. And we all use the same imaging platform, same version. This is the TomTech. We use a TomTech image analysis software. Why? Because it works well with all types of echoes from different vendors. We analyze the echoes in DICOM format, and TomTech is a good and FDA approved, etc. Software. May not be the best, but it works very well for us. So the sonographer will do all the quantitative measurements, the tracing of the gradients, the valve areas, etc. Then the echoes are, with all the measurements and images, are transferred to the cardiologist for overread. So the cardiologist will typically check all the measurements, eventually correct some of the quantitative measurements done by the sonographer, and will do all the qualitative grading. So all the qualitative assessment, the grading, in particular of the aortic regurgitation, mitral regurgitation, identification and description of the potential complications are done by the cardiologist. And then we have a last step, that is me. I'm overseeing and double check everything. I'm a kind of a bit obsessive compulsive with the echoes, and I like to have a final look on all the data and everything. And sometimes I do some additional corrections and modifications to justify my salary, I guess. And so this last validation, once it is done, then the echoes are saved in a server and backed up in 2 different servers in 2 different locations with all the measurements. And what we do is we directly enter our measures in an electronic CRF. We don't use any more paper CRF. So typically the sonographer will fill the fields in the CRF. Then the cardiologist check this data and eventually enters new data. And same, I am doing a final check at the end, and then I'm going to lock typically the CRF. And I think you're going to have the presentation on the CRF next, but the CoreLab will be one of the eCRF. So if we move to the next slide, and I may go more rapidly on the other aspects because it might be a bit more specific and technical, but what is the objective of the echocardiographic imaging protocol? Of course, this is to obtain an initial echo Doppler examination at baseline to assess the cardiac anatomy and valve function. Of course, we're going to have a particular emphasis on the aortic valve, the left ventricular size and function. And then if we go to the next click, the primary goals of the follow-up, so post-implant studies, post-AVR, is to, of course, assess the valve device, its location, function, and to determine how the AVR impacts the cardiac function. So if we go to the next slide, this is the imaging protocol, and this is the schedule of the echo visit. You should see a small table, and actually the table shows that we're going to collect, in the context of the Rheia trial, we're going to collect the baseline, of course, in all patients, then the discharge echo, essentially for safety purposes, and then the 30-day. 30-day is a very important one because generally the quality of the echo is better. This is the baseline post-implant function that we can assess at 30 days, and then we're going to do the 1 year. The window range acceptable for the that I put here are probably not final. This will be eventually adjusted in the protocol, so stay tuned. So I actually put the time window that we generally use for the partner trial. So for the baseline, we allow a 3-month window. So if you do the echo within the 3 months of the intervention, this would be acceptable to us. For the 30-day, we allow a 7 days window around this time, this date of the 30-day date actually, and for 1 year it's a 30 days window. Maybe the 1 year is somewhat strict, but we'll see. So if we go to the next slide, I just want to emphasize the most important thing. We want to use, of course, good state-of-the-art ultrasound system. The only accepted format for the echoes to be sent to the CoreLab is a DICOM standard digital. I mean, it's pretty standard, but we cannot, of course, read AVI files or native files from the manufacturers. We need the DICOM. And the next click, please. Actually, this is just to show what is the frequency range that is typically accepted, but this is generally standard. So the next slide, you can go on all clicks, please. There are 3 clicks, so we can see the 3 paragraphs. Of course, we would like to have a qualified physician or sonographer to do the ultrasound exam. Ideally, if you can use the same team for the different echoes at baseline and follow-up in a given site, this is always better because there is always a viability related to the sonographer or the person doing the image acquisition. This is not mandatory, but this is ideal. So ideally, same team and also same system. Because if you change, if you go from a GE to a Philips, it might also introduce some viability. This is not mandatory again, but this is best. We can go to the next slide. So one of the things that is important when we do the echo is to have a high quality ECG. I still receive some echoes with no ECG or very low amplitude ECG. ECG is important for us to synchronize our measurements. So please, this is an important thing. There should be no patient personal identifier visible in the header or of the echo file or the images. So we want to be sure that the echoes are well anonymized. Generally, the company that ensures the image transfer also provides to the site a platform that allows anonymization. So this is, of course, a very important thing to protect personal information. If we go to the next click, generally, I don't know if it's going to work that way, but we of course, we need to identify the echo using a study ID. That will be a labeling of the echo according to the site number, patient number, and visit that will be provided by the coordinating center. So if we go to the next slide, it's kind of busy. You should see two tables. Actually, this is the image acquisition checklist that we will provide to the site. It's a two-page sheet that the sonographer or the cardiologist doing the echo can use to check to make sure that they provide all the views and the signals and images. It looks like a lot, but in fact, you will realize that this is a pretty standard, routine, complete image acquisition that we do for a patient with valvular disease. So there is the parasternal long and short axis, the RV inflow and outflow view, the parasternal short axis, and then the apical 4-chamber, 5-chamber, 2-chamber, 3-chamber views, and then finally the subcostal and all the right parasternal and suprasternal notch views. So, I mean, this is the usual imaging sequence that we use, and there are a few specifics and things that we request in the trial, and I will cover them. And anyway, in the webinars that we'll give for the sites later on, that generally is very well described. So if we go to the next slide, I think one of the important points with regard to assessment of regurgitation in particular, and particular aortic regurgitation, because one of our, of course, objective is to ideally demonstrate very low rate of paravalvular regurgitation in this population with the Sep2019 compared to TAVR. A multi-window imaging is key with color Doppler. So the four main views are presented here: parasternal short axis, long axis, and the apical 5 and 3. But more views we have, more happy we are. And this is for AR, but also for MR. So color Doppler interrogation in multiple views, multiple planes. If we go to the next slide, one view that is extremely important is the parasternal long axis view with a good view of the left ventricle and a focused view, zoom views on the aortic valve, of course, with 2D with color Doppler. This is one of the key views where we do the LVOT diameter measurement, which is always a challenging measurement and key to calculate the valve area. If we go to the next slide, this is the apical 4-chamber, which is also extremely important. We need good views, 4-chamber with a zoom views on the left ventricle for assessment of LV ejection fraction and also different parameters that we can measure, especially the right ventricular function. And that's one of the things that we discover with the Partner 2 and now even more with the Partner 3, and I'm sure we'll see in the Rheia trial as well, is that maybe the crux and the most interesting information is from the right ventricle. And when we compare TAVR versus TAVR in Partner 3, there was not much difference in terms of recovery of LV function, LV ejection fraction, and even global longitudinal strain. But when you look into the right ventricle, this is where you have a big difference. The RV function is dropping with TAVR, whereas it is relatively well preserved with TAVR. So I want to be sure in this trial that we also collect this information regarding RV function. So apical four-chamber optimized for the right ventricle is important, and we're going to measure in this trial, as we did in the partner 3, we're going to measure the TAPSIR, but by 2D method, because I realized that when I asked the sites to do the TAPSIR by M mode, in about 50% of the cases it's missing. Whereas the TAPSIR by 2D, I only need the standard four-chamber view with some optimization for the right ventricle, and I'm able to do this measurement in the vast majority of the patients. So, and it's a reliable parameter that actually predicts mortality in partner 3, as we presented at the late-breaking trial activity. So the next slide, you should be on apical 5-chamber view. Is that right? Maybe if not, well, advance until you see apical 5-chamber. This is a very important view as well, because this is where we're going to assess the AR and, more importantly, measure the velocity in the LVOT. So if we go to the next slide, so for this, we're going to ask the sites to do sampling of LVOT velocity just below the valve and just in the stent but below the leaflet. This will be well described in the protocol, so no worry. And it is also mentioned in the checklist. So if we go to the next slide, that should be apical 2-chamber view. Same recommendation of 4-chamber. We need a good view zoomed of the left ventricle, because this is a view that we use for the biplane Simpson. And LV ejection fraction is always an important parameter, so we want to make sure we have a good 4 and 2-chamber to do the biplane. If we go to the next slide, this is the 3-chamber view, okay? That's the last of the apical. And it shows here, well, the two sampling of velocity, because the velocity that we do by pulse wave Doppler, we request in 5-chamber, but also in 3-chamber. So we have more signals. We need actually more acquisition we have, and with many beats, not traced is good for us. And so it is shown here what we should do for TAVR and for TAVR in terms of sampling of LVOT velocity. And if we go to the next slide, and that's an important one, that's the right parasternal view. Why is it important? Because in up to 50% of the patients, you will find the maximum transaortic velocity at this right parasternal view and not at the apical views. And many sonographers and cardiologists, they sometimes do the apical views and they forgot to do the right parasternal. So right parasternal is extremely important to acquire, so we are sure that we did not underestimate the gradient at baseline or at follow-up, the aortic gradient. And if you go to the next slide, this is exactly what I mentioned. It's the a study showing that you will find the maximum, the highest peak velocity and gradient, not at the apical chamber, but in the majority of the cases will be a right sternal border, okay? That's a study from the Mayo Clinic, actually. And if we move to the next slide, this is an example that actually I got from a patient who was recruited in early TAVR, and it's the same patient with different views. So my sonographer first said, well, this is not severe, the patient is not eligible for the trial. The velocity was 2.9 meters per second, but this is from the apical view. But she did not at the time interrogate the right sternal border. And when you look at the right sternal border, the conclusion was much different. The velocity was 4, and then all of a sudden, the patient was asymptomatic because it's for early TAVR, was eligible for the trial. So I think it's very important to, so I insist a lot when I'm giving the webinars for the sites on this multi-window interrogation with continuous wave Doppler. So if we move to the next slide, this is the last part, how to transfer the echo images to the CoreLab. I don't think, well, this slide is any more relevant because I put the slide that I generally put when we use BioClinica as the provider, but I think we'll use a different provider, so probably it will be a different platform and different template, but generally it's always the same. The site has to enter the identifier of the echo, and there are a few questions generally that are requested. So there is generally a transmittal form that is associated with the echo that will be uploaded. So again, the echo needs to be anonymized and in DICOM format. And if we go to the next slide, we have a quality assessment process. So we actually, for each echo, we look if the echo acquisition was complete, and if there was some missing images or some acquisition that was suboptimal, and we provide feedback to the site. Generally, we do this feedback via the image provider, the image company. We could work differently if you want in this trial. I'm flexible. It needs to be simple process though, and sometimes we have some queries that are more urgent. For example, if sometimes we receive echoes that have been, maybe the file has been corrupted, and so for example the video clips are not playing. I have only fixed images. So then I have to go back to the site and say, can you re-upload the echoes, and or do you have any more images? Or so we do a rapid query in this case. So and then the next slide is just to thank you and know if you have any comment or suggestion or question. Thank you very much.
Thank you very much, Philippe. This was a very described and thorough description of the ECHO requirements. Personally, I'm not an ECHO specialist, but I have one simple question for you. You just mentioned that the right parasternal view was one of the best to assess the gradients, the peak velocity. Has it to be repeated during the follow-up once the prosthesis is in place, whether it be a transcatheter or surgical? Do we have to promote the use of that right parasternal view?
Yeah, absolutely, absolutely. We need to have systematically this view. So interrogation, especially the apical. So essentially for continuous wave Doppler, the minimum is apical 5-chamber, 3-chamber, and right sternal border. Because I've seen some cases, for example, after implantation, where you think, or you say, well, I have the 1-year echo, or the 30-day echo, and then the 1-year echo, and then the gradient has increased by sometimes 10 millimeters of mercury, and you say, oh my God, you know, it's maybe a subclinical thrombosis or something. But in fact, you realize that this is just due to a difference in imaging. At 30 days, we only have the apical 5-chamber or 3-chamber, no right sternum border, and then at 1 year now you have the right sternum border and you have a higher gradient than the apical. So that's why, yeah, it is indeed very important to be sure that we have this right sternum border, not only at baseline, but also at follow-up. And eventually, maybe in the ECHO-Corelab CRF, we should maybe have a checkbox where we indicate which is the view that we use to measure and report the gradient.
Could you confirm to the investigators and the coordinator that they will have a kind of booklet, a manual to follow all these rules and to do it properly?
Yes, so there will be several things. Generally, we provide actually three main documents. One, I think, which is the most useful, is similar to what I presented today, but it's a more comprehensive PowerPoint. I already sent it to Peter and Lenka, so it's a real detailed PowerPoint presentation, because when you talk about imaging, it's better to have images to show what we need in terms of acquisition. So I'm covering the complete protocol, the imaging sequences, and the tips and tricks. And so I can update this PowerPoint presentation, and I think it would be good that this PowerPoint presentation be provided to all the investigators involved in the performing of echoes. There is also the checklist that I showed that I think is useful for the sonographer during the realization of the echo. And we also have a manual where there is more text than in the PowerPoint, and where we actually, again, put a lot of images so it's a kind of a manual of operation for the image acquisition protocol. And of course, I did not mention, but all our measurements are standardized. The image acquisition needs to be standardized, but also our measurements are extremely standardized. So we have, for the ECHO-Corelab, we have standardized operating procedure, SOP that we follow. So we all measure or try to measure the same way. But I think this information will not be transmitted to the site because they are not, they do not need to do the measurement. What I want to be sure is that they have the complete information for the imaging acquisition protocol.